Welcome once again to our YouTube channel. Thanks for subscribing to our channel. We really appreciate you. Today we quickly want to look at a particular topic in biology, which is genetics. What is genetics? Genetics simply talks about the study of genes. So we want to look at majorly the crosses that are found in genetics. That is basically what we want to talk about today. So we'll be looking at the different types of crosses that we have in genetics. And suffice, we need to talk about genetics. So what is genetics? Genetics is simply talking about the study of genes. Basically, that is what genetics is talking about. Genetics. The study of genes. Basically, that is what genetics is talking about. So like I said, we are basing today's discussion on the crosses, different types of crosses that we have in genetics. So we'll be looking at the type of offspring that can be gotten from different types of parents. And now, for us to quickly talk about this, we need to understand some basic principles. We need to understand a few things before we quickly go into talking about the different types of crosses. First and foremost, I want to start with what we represent genes as. We have different types of traits that can be talked about when we talk about genetics. Traits like height, shape of head, skin color, length of fingernails, and a whole lot of others. All of these characteristic traits are depicted or they are represented with the use of alphabets, with the use of letters. So when we talk about traits or genes, we depict them using alphabets. So we can see something like this as an example of a gene. We can also see something like this as an example of a gene. So either of these two can be seen as a gene. We can also have another scenario that we have this. Now, when we look at genes, using different letters to depict them, they all mean different things. And so that is where we'll be starting from. First and foremost, let's look at this scenario where we want to talk about height as a characteristic trait. If we want to talk about height, let's assume we want to talk about height. You know, when we look at height, we'll be having tallness and shortness. So let's assume I want to depict my tallness with T. So I am using capital T to depict tallness. Now, the extreme opposite of tallness is going to be shortness. So let me put this here, shortness. So if I am using capital T as tallness, the extreme opposite is going to be small t, small t. Now, at this juncture, I want us to understand that whenever you are talking about a particular trait, it is very important that you know that you have to depict them with the same alphabet. The fact that I want to talk about height is the reason why I have chosen to use T. Now, for height, I will be using only T. I will not say because I want to talk about tallness, I use capital T, and then for shortness, I use a different letter. No, I will use the same letter. The only difference is going to be that this is in capital case, and this one is in lower case. This is in upper case, and this is in lower case. That is actually going to be the difference between them. So this is representing tallness. The capital T is representing tallness, while the small t or the lower case of t is representing shortness. And that means that in the middle there is a possibility of something that looks like this, where we have tallness and shortness, and that is of the average height. The person is not too tall, nor is that organism too short. So we have tallness, we have average height or medium height, and then shortness. So that is it. If I want to use this to depict something else, let's assume I want to talk about skin color. I could be using this as skin color. Let's say I'm using capital B as dominant black. It means this is black. And the extreme opposite of black, I'm using small b. Since I'm talking about color, this is black, this is white. So in the middle, I could be having something that looks like this, which is not is black, but it has a trait of white in it. So we could say for now let's call it white and black, or we can also call it hash. White and black or hash. Are we together? So basically that is what it is. So looking at this now, we will notice that here we have capital letters, here also we have capital letters. So whenever we see something like this, it helps us to understand that we have what we call the homozygous and the heterozygous trait. What do we mean by this? When we talk about homozygous trait, we're talking about a particular gene having both of its alleles to code for the same thing. When we look at this now, this is an example of a gene or a characteristic trait. Now, this gene, when segregation occurs, it splits this gene because we have two laws of Mendel. The first law of Mendel is the law of segregation, while the second law is the law of independent assortment. So when segregation occurs, it breaks, it splits apart this particular trait here into its complementary alleles. So this is an allele and this is another allele. Each of this is what I refer to as an allele. So when the two alleles in a particular trait, when they are coding for the same thing, when they represent the same thing, we say it is homozygous. Homozygous, or we can call it homologous. We can call it homologous when they are coding or representing the same thing. Example, we have capital T, capital T. This one also, where it is small t, they are both coding for the same thing. Each of these is an allele, and they are both coding for the same thing. It is also referred to as homologous. So when we look at capital B, or small b, they are coding for the same thing. So they are all referred to as homologous or homozygous traits. Now, when we look at this one here, this scenario where we have capital T, small t, this particular one that I'm staring, where we have capital T, small t, or this one here, capital B, small b. Both of these have their traits with different alleles. If you look at this now, we have capital T and small t. Unlike this one that we have capital T, or this one that we have small t. This one here is capital T and small t. As well as this one here, we have capital B, small b. Opposing to this, which is capital B, or this is which is small b. So these ones that are coding for different things, they are referred to as the heterozygous traits. This one here, we call them the heterozygous because they are coding for more than one trait. They are coding for more than one in that particular gene. So it is talking about, for this one, it's talking about color, but in that color we have black and we have white. For this one here, in this height trait, we have tallness and shortness. So when we have more than one, then it is referred to as the heterozygous trait. Now, from this heterozygous, we get to understand what we mean by the dominant and the recessive trait. In every heterozygous trait, like I said, we have two alleles. The complementary alleles that we have from this is going to be capital B. We having capital B and small b. We have capital B and small b. From this now, each of these alleles, this one here is coding for, we have it to be black, while this one here is coding for white. Now, in this particular scenario, we have the capital B overshadowing the small b. The capital B, which is black, will overshadow the small b, which is white. And so, the physical expression of this trait is going to be that it is going to be black. The physical, what you can see on the outside, it is going to be showing black, but it has a trait of being, or the possibility of being white. We have this as a recessive trait in this gene. As well as this one here, this small t, which is coding for shortness, is going to be a recessive trait in this gene because the capital T will be expressed, will overmask, it will mask the small t. So we have this organism to be done with the recessive trait of being short. So that is basically what homozygous and heterozygous trait talks about. So since we now understand this, let us move further by talking about the types of crosses that we can have. And so we'll be starting with what we refer to as breeding through. Breeding through. That is where we'll be starting from. Breeding In feeding through, we are actually talking about a cross where both of the parents have the same homozygous trait. That is basically what we talk about in breeding through. So we'll be having something that looks like this. We have this capital B reacting with capital B. Well, whenever we have crosses to make, there is this formula that I help students to understand, making their work neat. It is this: 1 by 1, 1 by 2, 2 by 1, 2 by 2. Looking at this now, that is what we use to cross this to make it easier. This is 1, this is 2, this is 1, and this is 2. Now, we have the first one there to be 1 by 1. So this is 1. We're starting with 1 by 1. And 1 by 1 is giving us this. We are having B. And then we have 1 by 2. 1 by 2 is giving us B, also capital B. And then we are done with 1 by 1. We're done with 1 by 1 and we're done with 1 by 2. We're going to 2 by 1 now. So we have this 2 by 1, which is also giving us capital B. And the next one is 2 by 2, which is also giving us capital B. Now we can see that our cross is very, very neat. And from this now, we can deduce that all of our outcomes, they look exactly like the parent. You can see all of the outcomes. Capital B, all looking like the parents. So here we have the parent, here we have the parent, and here we have the offspring. So we could also be having this possibility where we have small b being crossed with small b. In this scenario, what our possibility will be looking like is this. We have all of the possibilities looking like the parent organism. So basically that is what the cross of breeding through looks like. All of the offspring will look like the parent. You can see from this parent here, small b, we have all of the offspring being small b. So that is what breeding through talks about. And then we go to what we refer to as the single factor inheritance. Talking about the monohybrid. That is what single factor inheritance is talking about. We're talking about the condition where we probably cross this with this. So if we were to cross this with this, let's see the possibility of offspring that we will be generating from such cross. We are having this being crossed with this. We have it being crossed with this. Capital B being crossed with small b. So what is going to be the possibility of offspring that we will be generating? From that, we'll be having something that looks like this. We have capital B and then small b. Another cross like this. We also have capital B, small b. We also have another one like this, where we'll be having capital B and then small b. And lastly, we have this one. It's going to look like this, capital B and the small b. So looking at this now, all of the offspring are looking different from either the father or the mother. They all look different. We have capital B reacting with small b, and this is the possibility of offspring that we are generating from such. We are having all of the offspring looking different. Now we have homozygous and homozygous giving us a series of heterozygous. That is what we are having here. So permit us to say that from a cross where we have both parents to be homozygous, in this scenario we have this one to be homozygous dominant and it is green in color, while this one is homozygous dominant, but it is blue in color. So crossing these two traits now, we'll be having the green color with the recessive trait of being blue. We can see in all of the offspring, we have the green color with the recessive trait of being blue. That is what we can see. This is green with the recessive trait of being blue. So that is what the single factor inheritance talks about. Having understood what the single factor inheritance is talking about, then let us look at the crossing of the F1 generation. So I will see this. I will call this the parent, and I will call this the F1 generation. So when I cross the offsprings together, when I cross this and this together, let's see the possibility that we will be having from that cross. If I were to cross this, let me cross this and this. So we are having traits that look like this. We have capital B, small b, capital B, small b, and then capital B, small b. We have this reacting with this, so we'll be having from that, we'll be having this, capital B and capital B. That is what we'll be having from this. And then we have the next one, which is this reacting with this, 1 by 2 now, and that is going to give us this. We also have this scenario where we have 2 reacting with 1, and that is going to give us this as well. We have this, and then lastly we'll be having this reacting with this to give us this. Now, looking at the crossing of the F1 generation, we'll find out that we have an heterozygous trait for the father and the mother, heterozygous trait such that we have a dominant green color with the recessive trait of being blue for the father, and likewise the mother, dominant trait of green with the recessive trait of being blue. But from the cross that we now have, we have one of the offspring to have a dominant homozygous green color, and we have 2 of the offspring having green color. They have the same trait as the father, that is, they have green color. These 2, they have green color with the recessive trait of being blue. And lastly, here we have the last one, which is blue. It is homozygous blue because it has only blue that can be expressed in that particular trait. So from this now, we can actually look at what we refer to as the interpretation of this cross. So if we want to talk about the interpretation of this cross, we'll be having this. We have the capital green. This is talking about homozygous. This is homozygous green, this particular one. And in this one where we have this, where we have this, we have it to be heterozygous. This one is heterozygous, but green, because this capital B here, which is green, is going to mask the small b. So it is going to be heterozygous because we have 2 alleles here identifying, or we have 2 alleles here expressing 2 different traits. So this green, this capital B here is depicting green, while the small b is depicting blue. So that is why we call it heterozygous. And lastly, we have this one, which is small b, small b. And for the small b, they are both depicting the same thing, and that is why it is still homozygous. And this time around, what kind of homozygous? It is homozygous blue. That is what we are having in this homozygous blue. So now that we understand all of this, we can quickly express this. So what are we talking about? Looking at this, we can actually get out the genotypic ratio. So what we have here is the genotype. We have homozygous green, we have heterozygous green, and homozygous blue. Let me depict this very well so we understand it. So I said down is homozygous green, this one is heterozygous green, and this one, the last one here, is homozygous homozygous blue. So we can see, we can understand it better that way. So this is the genotype, and then we have the genotypic ratio. The genotypic ratio is going to look like this. The genotypic ratio is going to be, we have one of the homozygous grain, so it is one ratio. We have two of this, that's the heterozygous grain, and then one of the homozygous blue. So this is the genotypic ratio. And then phenotype now. The phenotype, we have green, and we have blue. Because this one here is green, this one is green, this one is green, this particular one here also is green, this one also is green. Because this B here, the capital B is green, and it is masking the small b. It's masking the small b, so we have this one to be green, and then the small b is just there as the recessive trait. So we have this one to be green, this is also green, this is also green. When we look at it from the outside, it is only this that is blue, so we have it to be, the phenotype is going to be green and blue. While the phenotypic ratio is going to be, we have 1, 2, 3 to be green, so we have ratio 3, and we have 3 ratio 1. So 1, 2, 3 is green, only this one is blue, so it is 3 ratio 1. So here we have our genotype, here we have our genotype, here we have the genotypic ratio, and then the phenotype, we have green and blue, and the phenotypic ratio is going to be 3 ratio 1. So basically that is what the crossing of the F1 generation, crossing the F1 generation. So when we cross the F1 generation, that is basically what we will be having. So since we now have this knowledge, and we have talked about the monohybrid cross separately, now we talked about the single factor inheritance and the crossing of the F1 generation from the single factor inheritance. Now we go to talk about all of this in the monohybrid cross. So this will take us to talk about the dihybrid cross, talking about more than one trait in a cross. So to discuss this, we will be using two traits. First and foremost, we want to use color and then shape. So color, shape. For color, I'll be using Y, and the capital Y is yellow, and small y is going to be green. And then for shape, I'll be using W as round and small w as wrinkled. So that is what we'll be using for our dihybrid cross. I said this is dihybrid. So this is what we'll be using for color. We are using Y, the capital Y is yellow, the small y is green. And then the shape, we are using capital W and small w. Capital W is round and small w is wrinkled. So we are going to bring out all of these letters to use them for our cross. Now we have capital Y, small y reacting with capital W, small w. So when we cross this is the possibility that we will be having. We have something that looks like this, capital Y, capital W, and then next we have this, where we have capital Y and then small w, and then we have this, where we have small y and capital W, and the next one we have this, where we have small y and small w. All of these are what we will be using in our dihybrid cross. Now, in dihybrid cross, we make use of a particular type of technique. It is called the Punnet'S square. The Punnett square was created by a particular scientist called Punnett. Now that is what we use in treating what we refer to as a dihybrid cross. So I need us to follow me as we will be talking about the crosses now. So we are having something that looks like this. So this is how the cross will look. The table will look this way. So we are going to put all of this now into the box. All of this is what we will be putting into these boxes to make our Punnett square. So let us start. First and foremost, we have capital Y and capital W. The next one, we have capital Y and small w. The next one, we have small y and capital W. And then the last one here, we have small y and small w. We are going to write the same thing that we have written here. All of this now that we have written here, we are going to write it here as well. So we have capital Y, capital W. We have capital Y, small w. We have small y, capital W. And then small y, small w. So it is with this now that we are going to create what we refer to as the Punnett square. Now we are going to be crossing all of this that we have here with what we have down the line here. What we have across will be crossed with what we have down the line. So let us start now. The first one we have here, we have capital Y from here, capital Y, capital W, and then capital Y, capital W. That is what we have from here. Capital Y, capital W, capital Y, capital W. The next one is capital Y, small w, and here capital Y, capital W. The next one is small y, capital W, and here we have capital Y, capital W. And lastly here we have small y, small w, then capital Y, capital W. So we understand how we did the first one now. We are going to follow suit for the others now. So let's write. We have capital Y, capital W, and then capital Y, small w. For the next one, we have capital Y, small w, capital Y, small w. For this one here, we have small y, capital W, and then capital Y, small w. For this, we have small y, small w, and here we have capital Y, small w. So we have this one to the capital Y, capital W, small y, capital W. This one is capital Y, small w, and small y, capital W. We have here small y, capital W. Here we have small y, capital W. Here small y, small w, then small y, capital W. And here we have capital Y, capital W, small y, small w. Here is capital Y, capital W, small y, small w. Here we have small y, capital W, and small y, small w. And lastly here we have small y, small w, small y, small w. So that is basically what our Punnett square requires of us. After doing this, the next thing to do is to number each of the boxes to identify them. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So these are the number of the boxes that we have. So with this now, we can actually go to write out our genotype. So what is the genotype going to look like? I will quickly erase this to make use of this place. So remember that we said that our yellow, our capital Y is yellow, small y is green, capital W is round, small w is wrinkled. So that is what we'll be using to express this now. Talking about the genotypes. So the first thing we have there, so number 1, we have capital Y, capital W. We have capital Y, capital W, capital Y, capital W. That is what we have as the first genotype. So interpreting that now, we'll be having this. Our capital Y is going to be brought together such that we'll be having something that looks like this, and then we'll be having this. That is basically what we will be having. So interpreting this now, not forgetting what we talked about earlier, this is going to be homozygous, and since they are both coding for the same thing, it's going to have the same color. So this is homozygous yellow because capital Y is yellow, and this one here, capital W, is homozygous also because they are both capital, and they are coding for the same thing, which is round. So that is how we all do all of the possibilities. So capital Y, capital W is going to give us, permit me to use this, HM here. HM is homozygous, while HT is heterozygous. That is what we will be using for this particular lesson. So capital Y is homozygous, and homozygous what? Homozygous yellow, and then we have capital W, which is also homozygous but round. Homozygous round. So why do we have them? We have them in number 1, so I'm ticking number 1. Number 1. Let's see if we can have them anywhere. We can have the same thing anywhere in all of these 16 boxes. Now I'm looking at all of these 16 boxes, I discovered that it is only in this number 1 here that we have that possibility. So it is, we are closing the bracket and we are writing 1. That is what we have for that. And then we move to the next one. The next one is capital Y, small w, capital Y, capital W. So this is what we are having. Capital Y, small w, capital Y, and capital W. That is what we have for that second one. So this is how we are going to interpret that. It is going to be homozygous as well. Homozygous, this time around it is also yellow. So we have homozygous yellow, and now we have this one to be heterozygous because this is capital and this is small. So this one is heterozygous, but the fact that this capital is here, the capital will mask the small. So the capital is round, so we have it this way. And where do we have them? We have them in number 2. Let us see if we still have it in any other place apart from number 2 in any of these 16 boxes. So we have it in number 2. It is not the same as what we have here because this one is capital, but this one is small. So it's not the same here. For this one, it is the same because we have it in capital, then capital and small. So we have it in number 2. Number 5, let's see if we have it elsewhere. So let's mark number 5. 5 is taken. So let's see if it is in any other place here. No. It is not seen any other place. So we have it to be 2. We have 2 possibilities of this particular one, which is homozygous yellow and heterozygous round. And then the next one, we have number 3, which is small y, capital W. We have small y, capital W, and then capital Y, capital W. That is the possibility that we have next. So what is the expression of this? This one is heterozygous yellow because this Y here is capital, while this one is small. That makes it heterozygous. Now this Y here, capital, is going to mask the small. So we have it to be heterozygous yellow and homozygous round. And homozygous round. So where do we have that? We have it in number 3. Yes. Let's see where else we have it. Capital and small y. It's not the same here. Capital Y. Okay, we have it here as well. Capital, yes, we have it in number 9. So 3, 9. Let's see if we have it elsewhere. Capital, no. So we have 2 possibilities of those as well. You can see. And then the next one we have is number 4. Number 4 is small y, small w. Small y, small w. And then capital Y, capital W. That is the next possibility that we have. So interpreting it now, we are going to be having heterozygous heterozygous round and heterozygous again. Heterozygous yellow and heterozygous round. This time around, where do we have them? We have it in number 6 is capital Y small y, capital Y small y. That is what our number 6 is. So expressing that now, we'll be having it looking like this. We have homozygous round and homozygous. This time around, it is not round, but it is going to be wrinkled. This one is homozygous yellow and homozygous wrinkled. Because what we have there is what? Double of the small w. That is what we have as our number 6. So number 6, let's see if we have it elsewhere. Capital Y, then small y. We don't have it elsewhere, so it is only number 6. So that is just one outcome. The next one that we have is let's mark 6. The next one is number 8. Number 8 is our next point. So we have small y, small w, small y, small w, and then capital Y and small w. That is what we have next for number 8. Now, that particular one is interpreted thus. We have heterozygous yellow, and the next one is we have homozygous wrinkled. We have heterozygous yellow and homozygous wrinkled. Let's see where we have that. That is number 8. Where else do we have that? Number 14. We have it again in number 14. So we have two possibilities of those. The next one we have now is going to be number 11. So number 11 is small y capital W, small y capital W, small y capital W. So if you look at this now, expressing this, since we have small y, it is not going to be yellow like we have had. This time around it is going to be green. And it is also homozygous. So homozygous green and homozygous round. That is what we have in here. So we have it in number 11. Number 11. Where else do we have it? Homozygous green. Okay, just number 11. Just number 11. And then the next one we have is small y, small w, then small y, capital W. That is what we have next. So looking at that now, interpreting it, we're going to have it looking like this. It is going to be homozygous green and heterozygous round. So that is what we're having there. And then what do we have? We have it in number 12. And then where else? Homozygous and heterozygous. So we have it as number 15 as well. Number 15. So we have two outcomes for that. And then we have only one that is not checked yet. The only one that is not checked is number 16. So that is the last trait that we are having there, which is small y, small w, small y, small w. So interpreting this now, we will have it looking like this. It is going to be homozygous but green, homozygous green, and homozygous wrinkled. That is what we will be having now. And the possibility we have is just for number 16, which is just 1. So all of these that we have written out now, they are the genotypes. So they should be 9 in number. All of these genotypes for a dihybrid cross should give us a total of 9 outcomes. So let's count. This is 1, 2, 3, 4, 5, 6, 7, 8, 9. So that tells us that we are on track. That tells us that we are doing right. Now, from what we have as the genotype now, you can actually get what we refer to as the genotypic ratio. The genotypic ratio. So what is the genotypic ratio going to look like? One of these possibilities that we have here is what our ratio will be. It's going to be 1:2, ratio 2, ratio 4, ratio 1, ratio 2, ratio 1, ratio 2, ratio 1. So if we count, all of this must give us a total of 16 since we have 16 boxes in our Punnet'S square. So let us see. 1 plus 2, 3, plus 2, 5, plus 4, 9, plus 1, 10, plus 2, 12, plus 1, 13, plus 2, 15, and plus 1, we have 16. So this here is our genotype, and what we have here is our genotypic ratio. And then, having understood that, we can now look at the phenotype and the phenotypic ratio. So the phenotype now, the phenotype. Now this time around, looking at the phenotype, we are not looking at it either being homozygous or heterozygous. All we need to know, or all we want to know, is the color and the shape. We don't want to know if it is yellow or green, or if it is round or wrinkled. That is all we are concerned with now, talking about the phenotype. So let us come to this. We have this here. We list out all of the phenotypes. So we have yellow and round. So yellow and round. So we have yellow and round. Let us see what else we have. We have yellow and wrinkled. Yellow and wrinkled. We also have green and round. Green and round. And we have green and wrinkled. Green and wrinkled. So we have all of these as the phenotype. We have some of them to be yellow and round. Some of them will be yellow and wrinkled. Some will be green and round, while some will be green and wrinkled. Those are the phenotypic possibilities that we have. The way we can see these substances outwardly, that is what we have, the four possibilities. Now, from that, you can now get what we refer to as the phenotypic ratio. So, the phenotypic ratio now. Phenotypic ratio. When we look at the phenotypic ratio, how do we get the phenotypic ratio? Let us look at all of the ones that are yellow and round. This here is yellow and round. You can see yellow and round. This also is yellow and round. This also is yellow and round. This also here is yellow and round. This one is yellow but wrinkled. This is yellow but wrinkled. This is green. This is green. This is green. So all of these ones, they are the ones that you have to yellow and round. So we add 1 plus 2, 3 plus 2, 5 plus 4, 9. So we have 9 as our yellow and round. Then the next one, we have the yellow and wrinkled. Let us see the ones that are yellow and wrinkled. We have this one here, yellow and wrinkled. This one also is yellow and wrinkled. Let's see. Okay, those two. So we have the next one, which is yellow and wrinkled. We have one. There are two phenotypes. So let's add the outcomes. 1 plus 2. We have 3. So we have 9 ratio 3. And then the next one, we have green and round. Green and round. This one also is green and round. This one is green and wrinkled, so this one is not part of them. So green and round, green and round. So 1 plus 2. We also have 3 for that possibility. And then the last one here is green and wrinkled, which is just 1. So that is what we have here. So looking at this now, talking about the hybrid cross, we have been able to prepare the Punnet'S square using these two traits, color and shape. And then we cross it to get all of these traits. in our Punnet'S way, and from this we interpreted this to get out all of these genotypes, and then this was the genotypic ratio that we got from the genotypes, and then from this we were able to write out our phenotype as well as the phenotypic ratio. So this is how far we can come, or as far as we can come in this particular topic. I want to believe that we are able to get one or two things from it. Please do well to go through it again. Try as much as possible to understand all that has been said and that has been discussed. And perhaps you have any question that you need clarification on, do not hesitate to send it to us on our comment section. We want to have your take on all that has been done so far. If you are yet to subscribe to our channel, please do not fail to do so. Subscribe to our channel, like our content and share them. Please, when subscribing, don't forget to put on the notification button so you can get easily notified when we upload new content. Thank you very much and God bless.